Everyone, and thank you for having interest in my talk. I'm a PhD student at Saarland University and at the Helmholtz Institute for Pharmaceutical Research, Saarland. And I am the chief developer of Nerpa 2, the tool which was released recently and which I would like to present to you today. First of all, let me establish some biological background. So natural products are a vast class of compounds produced by bacteria, fungi, and larger organisms. They often exhibit antibiotic activity and are involved in host microbe and microbe-microbe interactions, which makes them interesting both for medical purposes and for industrial ones. A notable class of natural products is nonribosomal peptides, and as the name suggests, they are not produced in the ribosomes, but have a more intricate biosynthetic pathway. Nonribosomal peptides are encoded in biosynthetic gene clusters, BGCs for short, but this encoding is indirect. A biosynthetic gene cluster directly encodes a large enzyme called nonribosomal peptide synthetase, and this enzyme in turn produces the final NRP product. The enzyme has modular structure and acts as a conveyor belt. Each module adds a new amino acid to the growing peptide chain and passes it to the next module. So it grows like this. And finally, the completed peptide is released and may undergo some additional modifications, such as cyclization, which you can see in this case. And the common task which natural products researchers face is to annotate a biosynthetic gene cluster with the nonribosomal peptide it encodes. And this task is actually very challenging for several reasons. First, this assembly line is not straightforward. Some modules can be skipped or iterated, and that's hard to predict. Second, it is also hard to predict which amino acids would be inserted by each module, and a module can insert non-standard amino acids, and also some modules exhibit so-called promiscuity when a module can incorporate different amino acids depending on the conditions. And at last, as I mentioned, the released product can further undergo some modifications, which also makes it not straightforward. So altogether, there are different weird things that can happen along the way. And in general, accurate predictions of a nonribosomal peptide solely from a biosynthetic gene cluster is impossible. However, we can still try to assess how well a given NRP matches to a given biosynthetic gene cluster and somewhat assess the probability that this NRP was encoded by that BGC. And this is exactly the purpose of the tool. So here we have the following problem. We are given a genome and some database of known nonribosomal peptides. And our goal is to try to annotate at least some of BGCs in the genome with the NRPs from the database. And this task is important for several reasons. For example, usually people are interested in finding novel nonribosomal peptides, and the usual approach for this is genome mining, and thus it is important to filter out the BGCs which are likely to produce already known compounds to save time. Also, this kind of task can be used in bioengineering when bioengineers try to find biosynthetic gene clusters which produce compounds close to the ones they need. So now, with the problem stated, let me introduce the pipeline of Nerpa 2. As the input, we have genome sequences and non-ribosomal peptides chemical structures, And the goal is to report the list of most probable BGC NRP pairs. As at the first step, we decompose each non-reversible peptide into a graph of amino acids, and we do that with the Urban software. At the same time, we annotate biosynthetic gene clusters with anti-smash. We locate BGCs within the gene, annotate modules in the BGCs. We also use Paras software to predict which amino acids will be incorporated by each module. Then, given a graph of amino acids and a sequence of BGC modules, we align them against each other. We cut the graph into linear fragments and align these fragments against the modules. So we score each pair and report the best matches along outputs. Okay, so as the name Nerpa2 suggests, there was also Nerpa1. Nerpa1 worked in a similar manner. It had more or less the same pipeline. However, Nerpa2 has two major updates, which I will talk about. First of all, we introduced a new probabilistic model and improved our scoring scheme. So now, when we have a sequence of modules in a BGC, we construct a hidden Markov model for them, where paths in this hidden Markov model correspond to tentative biosynthetic pathways. And these pathways may allow such events as skipping of modules, iterations of modules, and incorporations of some extra amino acids, for example, coming from some unannotated modules. Then, given some sequence of amino acids coming from a linear fragment of an NRP, we find the most probable path in this HMM, which we hypothesize to be the most probable biosynthetic pathway, which yielded this particular sequence of monomers. So in doing so, we need to assess how well a given module explains a given monomer, and we do this with a log-odds score. From the Paras tool, for each module, we have a probability distribution of on amino acids, which this module is likely to insert. And we also computed somewhat default frequencies from the database of known non-ribosomal peptides. And to score a match of a module with a monomer, we compare these two probabilities. The probability that this monomer was inserted by the module versus the probability that it just occurred on that position by random chance. Okay, so this improved model gave us better performance. We benchmarked Nerpa2 against Nerpa1 on about 140 BGCs from the MIB database. And we run Nerpa1 and Nerpa2 on them against the combined databases MIB and Narin, which have 750 NRPs in total. And then we counted how often it was that Nerpa2, the best hit for Nerpa2 was correct, and the correct compound was among the first two best hits for Nerpa2, among first three hits, and so on. And we did this the same for Nerpa1, and as you see, Nerpa2 is consistently better. The other notable improvement is that now we have an interactive graphical output. The users can view all the matches, sort them by score, by genomes, and by nonribosomal peptides. And also for each match, users can see annotated modules in the BGC and monomer graphs. And well, yeah, I'm out of time, I guess. The tool is available on the GitHub.
Thank you very much. Do we have a question? Okay, well, thank you very much for providing this interesting tool. And then I guess we move on to the next speaker.